From the vast array of evolution within infectious disease, we're going to move on to looking at development. So when a biologist talks about development, they're talking about the amazing things that happen when you have a single cell, which is a just fertilized egg, that can grow and make shape and structure and form. So for us, humans and other mammals, this mostly happens during pregnancy, and once you have a newborn, not much happens later. So, you know, quite a bit happens during adolescence. So for any teenagers in your life, give them a break. But there's a lot going on there. But, you know, we know that, you know, if you have an accident and you cut off the top of your finger, it's not going to grow back again. You know, that process has finished, that process of development. And that's one of the reasons there's a whole challenge in terms of regenerative medicine. It's very hard. But plants are very different. Plants have a much more plastic development. You know, you can cut a branch off and a new one grows somewhere else. You can have a leafing branch that can become a flowering, can have a flowering structure later. Any part of it might do this. And recently in my garden, I accidentally uprooted, exposed a bit of root of a plant of mine, and then a new stem just grew out of it. So they're very, very plastic in terms of development. And our next speaker, Ottoline Leyser, has had a wonderful career uncovering and discovering how plants can have such plastic development. And in the course of her career, she has discovered a lot about how the plant hormones can regulate development like this. So we're very lucky to have her here today. She's currently professor in the University of Cambridge, where she's director of the Sainsbury Laboratory. And last year, she was made Dame Commander of the Order of the British Empire, OBE, and is also a Foreign Associate of the US National Academy of Sciences, which is a special honor because they don't tend to let there's a small number who are non-US based. So we're very lucky to have Ottoline here today, and so I very much look forward to her talk. Thank you.
Thank you very much for that kind introduction. And as a number of the other speakers have said, it's really a joy and a privilege to be here at this very exciting meeting, thinking much more globally about what is life. And I feel particularly a kind of burden of responsibility as the representative of the plant kingdom here, because the plant kingdom and its photosynthetic microbial kind of relations, I suppose, is the point of entry into life, into the system of life, of all that negative entropy or free energy that Schrödinger talked about. And so it's quite an important part of that tree of life that we've seen, because without it, there is none of that negative entropy in the system, and everything else wouldn't happen. So it's kind of important, and so it's nice to be here and able to speak a little bit about that. And I think for me, this has always been quite well encapsulated by this experiment. So this experiment comes up a lot if you're studying biology, because it's quite often billed as a kind of important point in the history of science in general, because it's the kind of restart, I suppose, of actual experiments. And it was done by this guy, Jean-Baptiste van Helmont, and he sets out a kind of experiment to try to work out how it is that plants grow. And what he says is that he took this earthen pot, he carefully weighed how much earth he put into it, and it was dry, so he knew that that's all there was. And then he watered it with rainwater, and he put this willow plant into the pot, and he weighed that very carefully. And then he waited for 5 years, because he wasn't under the same grant pressure that the rest of us are these days. And after 5 years, he weighed it again, and he discovered that it weighed quite a lot more than it did previously, whereas when he dried out the soil and weighed all that over again, it really hadn't changed very much. And so he concludes that all things vegetable arise directly and in a material sense from the element of water alone. Because as far as he was concerned, that was the only thing that he was kind of feeding to the plant. And we can look at this experiment and laugh at how hilarious it is that anyone could have come to such a stupid and wrong conclusion. But frankly, what plants are doing is so astonishing that it's not surprising that you would miss it in an experiment like this, because what they're really doing is literally building themselves out of thin air. Yes, they need that water and a few mineral nutrients coming out of the soil, but they are sucking out of the atmosphere carbon dioxide, and they are using sunlight energy to smash the carbon dioxide in with existing small chain hydrocarbons and build these long chain hydrocarbons, which are absolutely where this free energy in life is kind of stored in the longer term. And to hark back to the mitochondrial discussion earlier, that is fueled by sunlight, driven by the formation of ATP in chloroplasts now. So that very similar proton-pumping ATPase is doing exactly the same job as happening in mitochondria, is building that more temporary energy source, so to speak, that drives the ability of the plants to smash together those carbon molecules. So it's a completely astonishing thing that's going on here, and is very easy to miss in an experiment like this, where you think you're only putting in water, but now we know, fortunately, quite a lot more about it, and can think then, in the context of plant development, that is my field of interest, what that really means for plants. So if you think about this in the context of what every plant wants, a plant needs water and carbon dioxide, as I've said, and light and those mineral nutrients, and then, of course, everybody wants sex, so that we've also heard is a good idea for a variety of reasons. And in the past, evolutionarily speaking, in the early plants that were in water, all of those things were actually relatively easy to get. You slosh around in the water and they kind of arrive by diffusion, and in terms of sex, your gametes can swim about the place and find some other gametes, not so hard. But with the movement of plants onto land, getting all of these things becomes much more difficult. And so here are the kind of land plants of the present, and they have had to go with a much more complex body plan with serious specialization of parts to allow them to collect up those things. So there's the roots, which are collecting up the water and the mineral nutrients. There are the leaves that are collecting up the light and the carbon dioxide. And then there's specialized parts to allow you to do the sex in this rather more complicated environment that is much more air than water. So it's more difficult to move your gametes around safely. And this specialization has driven quite a lot of the important aspects associated with the whole way that plants lead their lives, the whole kind of life history strategies, driven substantially by the fact that the root part gives you instantly a massive constraint as a plant, because you are literally rooted to the spot. So this is a kind of instant dual problem. The first is that you can't move, so your environment around you is changing, but you can't get up and move to an environment that's nicer. If it's cold, you can't put on a jumper, you can't go inside, you can't go to find a place where the sunlight's better. You have to deal with whatever it is you've got right there. And you have to do it in a way that is robust to predation. So you're a sitting duck. You cannot run away from your predators. This vicious, marauding slug is a serious problem to you, which it would not be to anybody who had any ability to move at any kind of speed at all. So somehow plants have to solve this dual problem of living in a changing environment and adapting to deal with that, and doing it in a way that involves a robust approach to predation. And the way that they've done that substantially is by having this incredible flexibility of form, this amazing plasticity, so you can adjust your entire body plan according to the environment in which you're growing, and you do it in a way that involves no unique parts. So plants are fundamentally hugely modular in the way that they grow, and the number of those modules can be changed, the types of modules can be changed, but there are almost always multiple ones of those same modules, and that means if anybody comes and eats any part of you, there's always another one. And also, as we've already heard, you can grow more. So we've got this flexibility of form with continuous growth and development throughout the life cycle of the plant and no unique parts. So that's kind of illustrated here. So this is a cross-section through the seed of an Arabidopsis plant, which is the kind of standard lab model. And at the end of embryogenesis, which is in kind of human equivalent, the point when your baby is going to be born, all you have is an incredibly basic body axis. This is the top, this is the bottom, and this seed is going to germinate. These seed leaves are going to open up and start the process of photosynthesis off, and then the plant is going to build itself out of thin air in a way that's incredibly flexible and modular, and it's going to do that because at the top of the shoot system here and at the bottom are established these groups of stem cells, these so-called meristems, that then continue to divide and build the plant from the top and the bottom in both directions. So this is a tomato shoot apical meristem. So this is this little group of cells in a plant that's germinated and is actively growing. It's a dome of stem cells. It's making this spiral of leaves as it grows and develops. Cells are being pushed out into the periphery. Some of them are then selected to make leaves. And so this group of cells is literally building the plant underneath it by pushing cells out into that plant body. And so this amazing flexibility in the shoot system comes from the fact that there's this shoot apical meristem building the plant underneath it. But on top of that, entirely new shoot apical meristems are established in the base of every single one of those leaves that the primary meristem is producing, and each one of those has the same developmental potential as this main initial shoot apical meristem and can do exactly the same thing as the main shoot apical meristem. So then you can make a plant which has an entirely new shoot coming out of the shoot. So effectively you have a kind of fractal system where the plant gets to pick from this incredible range of phenotype space from one unbranched shoot to an incredibly ramified bush, merely by deciding how many of these meristems to activate and when to activate them and where to activate them. And that's what gives you this incredible flexibility of form, and it does it in a way that means there are no unique parts, so that if any of those parts is eaten, doesn't really matter very much. And in fact, this control of which of these buds activate is something that's probably very familiar to anyone who gardens, because it's the basis for pruning. So if you come with your secateurs and chop the main shoot off, that's one of the things that will activate any dormant buds in these lateral, in these leaves here, and that's why pruning makes your roses bushy. And effectively, that's a predation response in a real plant situation. If your primary meristem's eaten by a predator, you've got spare ones that you can replace the shoots that you've lost. So that is all well and good, but what it means is that plants are then constantly having to make decisions about where to put the resource that they have. So they're doing photosynthesis, they're making all that lovely carbon in sugar in beautiful ways with sunshine, but what should they do with it? Where should they put it? They have this amazing flexible growth habit, and they could make more branches, they could make more roots, they could make more leaves. How do they make decisions about where to grow in a sensible kind of way? And we know that they do that, and we know that those decision-making processes are really quite sophisticated. So one of the main things we work on in my group is the control of shoot branching, and that is illustrated here in the context of nitrogen availability. So if you grow a little Arabidopsis plant like this with lots of nitrogen fertilizer, it will make lots of these branches, whereas if you grow it with relatively little nitrogen fertilizer, it will make relatively few branches. And you could argue that this is a rather trivial response, and it's just to do with whether you have enough nitrogen to build the branches, but we know that that's not true, and I'll show you in a minute the next slide, which kind of rather obviously proves it. But actually what's going on is that the carbon resource that is available to this plant, rather than investing it in making more shoot and more branches, is being shuffled off to the root to invest it in making more root, because if you're limited by nitrogen availability, the place where you should put your current resource is in the root, because that's where you're going to get the more extra nitrogen from. So this is a sensible decision on the part of the plant in that it's able to then prioritize its current resource in a way that will maximize its currently rate-limiting resource. And that adjustment is happening continuously all the time throughout the life cycle of the plant. And we've begun to learn something about the regulatory systems that allow plants to make those decisions. So I'm a geneticist, and the way we've done that in Arabidopsis is through a classic forward genetic approach. We look for mutants that don't do those decision-making processes correctly, and this is one of the ones that we've worked on a lot in my group. So you make one DNA base pair change, essentially. We've spent a lot of work figuring out which base pair that is. These guys and these guys differ by this one base pair, and these guys are incredibly branchy. They're even more branchy than the normal plants on high nitrogen, and they don't get any less branchy when you put them on low nitrogen. They're struggling a bit because they're trying to build loads of branches with very limited nitrogen availability, but they have made a lot of branches. And this absolutely demonstrates to me very clearly that this plant is not branchy because of a evolutionarily built regulatory system that prioritizes root growth over shoot growth under nutrient limiting conditions, which we have broken by making this one base pair change. So, just to emphasize this point, here's Darwin, here's the survival of the fittest. Natural selection in plants has done the same thing that it does in all living organisms. It works to maximize the number of grandchildren in the face of competition in this unpredictable world that we've just heard about in the context of microbes and their interactions with their hosts. And in the plant context, this results in this very flexible growth habit, all kinds of things to defend yourself, both in the context of the way that you grow and develop, but also you've got indigestible poisonous seed that you have to have cunning ways to disperse the seed from the plant, for example, and you want your seeds to be minimally resourced in a way that will allow the maximum number of them to germinate and survive and thrive in the next generation. And that's all very good, but this is actually in stark contrast to what you want in agriculture. If you want to use plants to make food, to provide negative entropy to the human population, for example, this list of things is really not a very good idea. And Darwin also spent a lot of time on domestication, on human selection, which you could characterize as survival of the favorite. And here, you want to do something altogether different, which is maximize the resource allocated to the part that we want to eat, which quite often is the seed. We're not really interested in the plant being able to compete with other plants because we're going to grow them all in a nice field. We really don't want them to compete with one another. We want them all to play nicely together, so no competition, please. And we're going to provide the resources in a way that we can control so you don't have to worry about, if you're a crop plant, the incredible vagaries of the environment, at least not to the same extent as you do if you're a wild plant. So there you want a whole bunch of different characteristics. You want a less flexible growth habit. You want the seed to be delicious, nutritious, and digestible, which is the last thing that you want in a natural selection in regime. And you would like the seed to stay on the plant so you can harvest them easily, and you would like as much goodies as possible put into that seed. So have we succeeded in doing this? We've done really quite a good job, I would say. So this is teosinte, which is the wild relative of maize. This is maize, which we know and love. And this is what natural selection has done, and this is what human selection has done. This is great if you're a plant living in the wild in Mexico. This is rather better if you're trying to feed a lot of people. This has taken about 8,000 years to do, and we'd like to speed things up a bit, please. So the way you could look at it is that a natural selection has given us a washing machine. It's got lots of very useful features. It's a nice white box. It squirts hot water on the thing that you want to wash. This is great. But on the other hand, if you want to wash dishes, it's a little bit of a problem because if you put your dishes into the washing machine, they're not going to come out in a very good state afterwards. So what we would like to have is a dishwasher. We want to keep the water squirty and all that kind of stuff, but we want to replace the rotating drum with these rather nice slidey drawers instead. And 8,000 years of domestication, we've done quite a good job on a lot of that. But these days, we need a super efficient dishwasher that uses less water, less energy, and less detergent, and also heats the kitchen. And so we kind of need to up our game a little bit on this front. And to do that, rather than 8,000 years of careful trial and error, as we heard about yesterday, it would be much better if we really understood how the washing machine works, how the dishwasher works, and therefore what targeted interventions we can make to tune these systems to be more productive and to do the things we need them to do in agriculture in the 21st century. And that's where understanding really what this one base pair DNA change is, comes into play, and we need to be able to do that. We need to be able to do that effectively. And I think that's what the future of plant biology is really about. It's about understanding, really understanding how it works. And what we've done here, as I said, was make mutations that have shifted the whole position of this system into a slightly different place. This is kind of like that grain of sand that we heard about yesterday. We've changed this one base, this one grain of sand, and that shifted the entire system, which has lived its whole life without this one base pair different, into a different place, which is this compared to this, and we need to understand how that's worked. Now, we've spent, I suppose, the first chunk of my career, the real bottleneck in understanding was figuring out actually what that one base pair was and what that meant for the organism in context of what the gene that we'd changed that was part of that one base pair was doing. In the context of the plants I've just showed you, it turns out that this is a gene that is transcribed and translated into a protein, and that protein encodes an enzyme, and that enzyme oxidizes this thing to this thing, and this thing is actually then further modified to make a small molecule in a class called strigalactone, which is a plant hormone, and that is tuning the degree of shoot branching. And this is nice, and yeah, so the difference between these plants here and these plants here is these ones can make this and these ones can't. And that's okay, but actually we're still miles away then from understanding why, how. The scales that we're talking about are completely different. And again, this harks back to quite a lot of talks over the conference so far that talk about this problem of trying to understand how things work, how information flows across scale, how you can get from this nanoscale right up to these centimeters of plants. So just a quick to illustrate the problem, this is the one base pair change here in some DNA, which is resulting in the production or lack of production of this nanoscale compound. But the scales we're interested in influencing, how branchy you are way higher than that. So, 1 order of magnitude up, we've got these chromatin fibers from that DNA folded. And then this is a picture of the outside of a nucleus where that DNA is with a nuclear pore. And here's the nucleus in a freeze fracture. And there's one of those chloroplasts that's doing the good job. And we're keeping zooming out 1 order of magnitude at a time. Here are some cells on the surface of a leaf. Here are the cells a bit more zoomed out. Here is the leaf. And here is the plant, and we've gone across 7 orders of magnitude, and to put that in perspective is more than 1 meter to the size of the Earth. So somehow or other, we have to understand how information is moving across that incredible range of scales, how that one grain of sand shifting, that one base pair change is moving that whole system into a different place. And we've done okay, I would say. I'm not beating ourselves up too much over the progress we've made, but it's not satisfying yet for a reason I'll get to shortly. So here we are, ramping our way up through the scales. Here's this molecule. We know quite a lot about the different proteins in the cell that you need to notice how much of this molecule you've got. And we know quite a lot about what those proteins then do as a result. And one of the things they do is to put different amounts of this protein on the surface of the cell, and this protein is involved in moving a different hormone around. So you can think of this cell as a dynamic, self-organizing system of interacting molecules like this that's sensitive to local and systemic inputs of various kinds. So we know that the amount of this hormone and some of these proteins are changing depending on the environment in which the plant is found, and that's resulting in different amounts of this protein getting stuck on membranes in cells. And then when you think about these cells, they're all contributing to things like the meristems that I showed you. So you can think of this meristem as a dynamic, self-organizing system of interacting cells that's sensitive to local and systemic inputs, and the amount of this other compound that's being transported about by this protein that is labeled here on the cell surface, that's called auxin. That's very important in driving the patterning, the making of these leaves, and it's exported from the meristems out into the rest of the plant, and then you can think of the plant as a dynamic self-organizing system of interacting meristems as a result of each of them exporting auxin and telling each other what's going on. That's also sensitive to local and systemic input. So you've got this multi-scale thing going on with dynamics all the way through as key. And that's kind of the point, really. We've done quite a good job of figuring out what parts are involved in the system. Through classical forward genetics, we've identified a bunch of parts, and that's those molecular scale parts, and I think that's been the defining feature of the last 20 years of biology, is figuring out those molecular scale parts and what they are and what they do immediately, built on top of a century of proper biology, looking at organisms and tissues and organs and how those things might work together. So we're doing okay on parts. And naming those parts has been really important to us to understand how those, to be able to talk to each other about how those systems work and to build an intellectual framework where we can really understand this thing about the relationship between scales and so on. So we've had to name parts. It has some drawbacks because it constrains the way you think. If you think about something in parts, when the system itself has no idea it has parts, it's just doing its thing. But nonetheless, those parts are really key part of our intellectual tool set in trying to understand how the system works. So that's good. And all of the things I've talked about here are parts: cells, molecules, meristems, organisms. What is missing, I think, is an equally well-developed intellectual framework, vocabulary, for thinking about the dynamic self-organizing system element that runs across these scales. And I think that's the part that we need to work on most consistently over the next 10 years to develop the same kind of intellectual toolkit to be able to think about organisms at multi-scales in the context of dynamic self-organization. So understanding these dynamic behaviors is, I think, crucial to really understanding the relationship across scales between genotypes and phenotypes. And like the parts we've talked about, they're relevant both within and across scales. And the problem is they're not really as intuitive as parts because you can't just look at them, as some people have said in their talk, and say that's how it is. You can't. You can't look at them in an intuitive way. And we need to develop ways to build our intuition about those parts. So what I'm really interested in is, can we recast these dynamic behaviors as parts? So, and there's an interesting kind of parallel effort going on to recast the little regulatory networks you get that underlie some of those dynamic behaviors as parts, and that's driven by Uri Alon's lab in Israel. So he's gone about classifying all the little networks you can possibly make out of 3 different components, for example, and he's demonstrated that you can think about those at multiple scales, so transcription, synaptic connections, or in ecological food webs. And if you go and look for those things, you can find different ones of them disproportionately represented at all those 3 scales. So these kinds of little chains, little networks you can find in gene regulatory networks, in neuronal circuitry, and also in food webs. And this is, I think, a useful kind of paradigm for what I'm talking about, but it's not the same thing because these little networks are very useful, but they don't actually tell you what the dynamic behaviors are. They're not the same. The same network can have different dynamic behaviors depending on the kind of properties of that system. And just to give a quick example, the sort of thing I'm thinking about, things like biological switches, where you could have the same kind of network that gave you a switching behavior like this from one kind of stable state to another stable state, but you could have many different relationships between those 2 states. So you could get there very quickly, you could get there in a much more gradual way, depending on some input-output ratio. The switch could be really difficult to flip back in the other direction, or it could be really easy. And all of those kinds of properties come out of the way that the systems are wired up. And those are the things that build the kind of decision-making processes that I talked about in the context of both cells and tissues and organs in plants. Multiple ones of these kinds of dynamic behavioral modules put together in different ways to build overall behavior of the organism. So what I'm really interested in trying to address, and maybe this is one of these lasting things that could come out of this meeting, and it's been very exciting listening to all the talks over the last day and a half, because this theme does recur over and over again. Can we find ways to classify dynamic system modules, dynamic behaviors as modules, as parts, that we can then use to build our understanding and our intuition of how these complex systems work. And I think we need that to understand this kind of flow. We need that to be able to intervene and modify in a sensible way. And I think I would obviously like to stop there. But maybe we can, I thought there was one more slide, but there wasn't. Maybe the kind of groupings of people that have been at this meeting can start thinking about getting together to make that kind of parts list for those dynamic behavioral modules that seem to underpin all the systems that we've been talking about across the whole of life. Thank you.